It's okay.
We live stream the most slow tech way that you could possibly do it, which is with a camera phone.
Want to make a message for the people on YouTube that it's going to be late because my mom said there were 5 waiting and now it's down to 3 because on YouTube.
Oh, on YouTube?
At 5:25.
Oh, that would be a flip post that I made.
And mom
yeah.
said that the numbers are dwindling. So.
Yes. Well, thank you for
You're
that.
welcome. That way people will know they're at least at the right spot.
Yeah.
I know. We don't have meetings.
Okay.
Oh.
Make sure that our view. Oh, is it on already?
Yeah,
Can you plug it onto our, can you share it onto our, it stopped because I stopped it.
Okay.
I need to get in here. Okay. So how do I make this go to? I'm just trying to get my screen to not be sideways. Oh, that's it. Am I not sideways anymore?
Yep, you're good.
Okay. Can you post that to our Facebook?
I'll try.
I will try to. I'm sorry to move your worms. I'm sorry, worms.
Yeah, that's okay. We can.
Uh,
Most of this audience is. Let's start.
Yesterday. Yeah. So.
Oh. Yeah, kind of what they do.
Here.
Remember to plug it in now. That's your battery.
Well, it's charged. I think it'll be okay. Gonna go with that.
All right, welcome biology faculty.
Pretty much.
You know who supports their colleagues whenever we have events like this, and I have to just say, like, that's pretty awesome. So, yay to biology. welcome. I'm so glad to have everybody here. And I'm even more pleased to present Dr. Carnell, Lucinda. We had actually planned on doing this before the pandemic. Like we started downloading papers that Lucinda and students had written back in 2019 and 2020. We had talked to Lucinda about this. You even took us on a tour of your lab, and we got all these ideas for like ways to do this small exhibit. But then the pandemic happened and it became kind of impossible to do that kind of thing. And so we had to put the idea to sleep for a little bit, just like you do with worms. I guess you put them,
We
freeze them. That's right.
We put them in a deep sleep. Yeah.
Yeah, that's what we did with the exhibit idea. We put in a deep frozen sleep. And then we had to reawaken it again.
Sorry about that. Yeah. That's for the youth.
No, they just got a real surprise right there. The YouTube people. So this is what happens when you
All right.
go as low tech as possible to do lunch.
Yeah, I'll
So
step away from-
that's okay. So I just wanted to say like a few words of thanks. First off, the biggest thanks goes to Lucinda for sharing your research with us. To work with a museum on an exhibit like this it's not easy. We are lay people. We don't know the first thing about any of this kind of research. And so what it takes is for people like Lucinda to have the patience to be able to explain things to us. When we're reading through, maybe I'll just hold this during the talk. So when we're reading through, here, I'll just do this. Hey, when we're reading through, papers and things like that, we're coming across, terms like electrotaxis, for example. Lucinda has been patient and kind and has explained things in ways that have made them clear to us. And so a really big thank you to you, Lucinda. And the next big thank you goes to Stephen, actually. So Stephen Brandt over here is a museum studies student here at Central, anthropology major, museum studies student. And Stephen, whenever we had to put this idea into the deep sleep, was willing to go along this journey with me starting in fall of last year. We began reading worm papers and things like that, and getting some ideas for the exhibit, some crazier than others, some that were not exactly executable in a tiny window on central space like this behind us. But nonetheless, Stephen, you did such wonderful work helping me in terms of, design and conceptualization. But also, I just want to say, like, the text was written by Stephen. All the papers that you had contributed were read by Stephen. And so as an anthropology major, it was really reaching outside of our comfort zone for him and a comfort zone for me. And so thank you so much. And so can we just give both Stephen and Lucinda a big round of applause? So my last thank you, last but not least, is to the College of the Sciences. So Window on Central, the small exhibit case over here, was originally, I think it was supposed to be like a janitor closet of some sort, like a maintenance closet. And then eventually it became a space that was a little bit different. They got the idea to use it as a way to showcase science research that's being done here at Central Washington University. And I can't think of a better use of space than that. Actually, it's far better than keeping our brooms and things, although it actually does some of that too. Still, you just can't see that stuff. So, I really want to thank the College of the Sciences. That's our home at the Museum of Culture and Environment. We are so happy for the support that the College of the Sciences gives us. And without further ado, I want to turn things over to Dr. Carnell. Lucinda, thank you. Oh, that's me. I need to do that. That's my job. Yep.
There we go. Okay. So I want to thank everyone for coming to the Tuesday evening to listen to this, especially my colleagues, right? You always have me back, and nice to see a lot of other friends. So I'm going to talk about my journey with C. elegans. And here is a movie of one of these forms. So it's Cynorhabdites elegans. And it's called elegans because it moves in a nice, pretty sine way, in an elegant fashion. So that's where it gets its name from. And so before I kind of start this journey, and I will explain the title a few slides in about an inkling for the organism. And so I was going to discuss a little bit my origin story as a scientist. So when I first finished college or high school, I didn't know what to do. I liked biology. I pretty much liked everything, but I didn't know what to major in. And so my mom thought, neither of my parents had gone to college, but an engineer, maybe I could be a biomedical engineer, because if I like science, that would be a good job. So off I went. I found a program called Rensselaer Polytechnic Institute in upstate New York and majored in biomedical engineering. And it wasn't quite a good fit. I didn't really like how machines worked. I think the only class I liked was when I finally got to do a physiology class. So understanding all the physics behind biological systems made sense. And then I was also told at this engineering school that the half-life of an engineer was about 5 years and then you go into management. So I didn't want to be an engineer, and I certainly didn't want to be a manager. I mean, who wants to at like 19 or grade. So I decided to pack up things and go back home to Southern California, and I started at Long Beach State. And I majored in biology, physiology, and I got into a research program. And so before C. elegans came my organism that I have been with for 25 years, I had some other organismal partners. And so the very first one, research experience, was looking at barnacles. And so they're on the inner tide on the coast of Southern California. And this lab was interested in looking at what kind of protective proteins were expressed to sort of protect it from the heat in the environment. So they were in different ranges of the intertidal zone. So it was trying to get those barnacles, and if you know, they're stuck on the rocks pretty solidly. So I would just find chunks of rocks to take back to the lab and try to break these open and isolate tissue and grind them up. And I didn't really relate very much to that mentor. They just never seemed to be that interested in me, and I sort of hung out with the grad students. But then I decided I had two friends that were in the lab down the hall, and what they worked on, became my second organism, was a fungus called Palabas longicaulis. And so, is there any sort of pointer? Or
Yeah.
You can see those little spores on top, and they shoot off into the air, and then they germinate. And so what we studied
Should be the top one. Oh, we have to take the USB out of it at the bottom. Here. We love technical issues at the Museum of Culture and Environment. So
that's the little spore that we studied, and we studied their germination. And so we looked at what would cause those to germinate, and that was exposure to different sugars. And then that sort of activated a pathway inside this little fungal spore. And that was a great lab. I loved working with our mentor. He was a cowboy from Wyoming, Dr. Barrett, and really got my first exposure thinking I could actually do science and be a scientist. And it was during this time that I published my first paper, and it was in that lab. And so that was with Dr. Barrett, another colleague. And so that was my second organism. And then the summer between my junior and senior year, I went to a research program at UC Davis. And so there I had my third organism that I kind of hung out with, and that was a hamster. And so a Syrian hamster, you may not realize, is a hibernating animal. And so what this lab studied was what causes hibernating animals to awake. And so part of that has to do with the brown fat that Dr. Binder works on. And so I isolated brown fat from these hamsters, and I looked at a role of a protein involved in the activation of the hibernation of these animals. But I was studying that protein in isolated cells. And so it was in this lab that Dr. Barbara Horvitz that I really got an experience of maybe what a grad student experience would be like. So it was a great lab. There were graduate students, technicians, undergrads. It was a really exciting environment. I learned about different cultures and people. I learned to bike ride at Davis. And so it was just a great experience that made me think maybe I could go to grad school. the director of the program was Raymond Rodriguez, and he was a famous plant geneticist. And he had discovered this, or he developed the first kind of vector that you could put genes in, and you could kind of make transgenic organisms. And that first vector was called PBR 322, and he was the R. So he was like a famous scientist. But what was really cool is he would invite us once a week over to his house and have dinner and watch a movie. And so that's where I really felt like scientists were really kind of relatable, that maybe this was something that I could see doing. And so they were kind of my first mentors. And so at that point, that's what I did. And I went off, oh, sorry, I'm going to talk about this program. So at Long Beach State, I was in a program, and it was called Minority Access to Research Careers. And that's what helped provide resources for me to work in these research labs. And one of the things is we got to go to a conference at the National Institutes of Health, which funded this program. And so I found this brochure a few years ago. So it's a little raggedy because I pulled it out of a box, and this is me as an undergrad, and we're posing for this brochure for this program. And it's me and another classmate. And, you know, it's kind of telling I'm being very serious there, a machine that's not on. Here, we thought was kind of telling when I pulled this out. So I wanted to get a PhD in neurophysiology, and I ended up being a neuroscientist, although my degree was not necessarily in neurophysiology. But I remember at the time, I didn't like the quote. So it said, people had been so helpful to me that I wanted to put some of that back into the system, right? The other person had this cool quote about being a scientist, you know, and as a woman, it was like, oh, I want to be helpful. And I know that sort of bothered me at the time. But as I pulled that out and realized, right, I guess I didn't have very good self-awareness then, but that's exactly what I've been doing ever since I actually finished my undergraduate. So my first experience was when I got to graduate school in Berkeley, I became a tutor for an Upward Bound program, which is a TRIO program for high school students in Richmond, California. And then I became part of a group called Scientists of Color. And so one of the things we did is we worked on peer mentoring. We developed a first-year experience, and that has developed and still exists at Berkeley, and it's now part of a biology scholar program, which has really been transformational. And then the other thing is we did a distinguished minority lecture series because we were told as graduate students there were diverse faculty at Berkeley because there were no faculty of color doing good science. So we started an annual lecture series, finding them for them to show. So anyway, it's kind of funny that ended up basically what I have been doing for the next 30 years. So I went to graduate school at Berkeley, and I still didn't find the worm. I found culture cells. So I grew cells in culture. And what I studied was how proteins like insulin get packaged and secreted from cells. And so this was my first paper I published as a grad student, and this was my mentor, Xiao Ping Moore. And one of the things that was really amazing about her as a mentor is at one point, I was always trying to just do experiment after experiment to show how hardworking I was and devoted. And she actually said that I would be better off maybe not doing as many experiments, but just really thinking about one good one. And so I thought that was really important. She kind of developed me as a scientist and my intellect. And she was also very supportive of my involvement in that program for sort of me and my identity. And so she was a great mentor. So now I will get to my first why C. elegans and how I discovered C. elegans. And I didn't discover it, but how I discovered an interest in it. So I went to a talk at the end of grad school by Josh Kaplan, and he was using this microscopic worm, C. elegans, to study nose touch. And so if you kind of look at this right at the bottom, it's literally, it bumps into a hair and then it reverses. So here's a behavior. You have this organism that has a really easily studied behavior. And then I'll talk about this a little more detail, but we know every cell in this worm, so we know all the neurons. So he knew all the neurons, the sensory neurons that detect the touch. And then we know the neurons they connect to, and then they connect the motor neurons that connect to muscles that mediate the behavior. So I can study the behavior, the physiological system, an individual cell, and through the awesome power of C. elegans genetics, you can identify genes and proteins involved in this process. And so what he discovered is he looked at mutants that couldn't respond to touch, and he identified a glutamate receptor. And we have a way to visualize cells by making them green in the worm. So this is what you see here, and that's that receptor. Okay. And so that's what I was really interested in. So for my postgraduate work, so after a PhD, you still go do more research and training in science, and or at least in our discipline. And so I interviewed in the Kaplan lab, and he actually didn't take me. So, but you know what I realized? I'm not the only one in town that studies touch and smell. So I did also interview, I don't have his name up there, Dr. Martin Chalfie, and that was at Columbia University. So I went to Columbia University, New York, and we studied gentle touch. So that eyebrow hair, it goes across the head, it reverses across the tail and moves forward. And so these are the identified neurons that sense that. And so I'm not going to go into that project, but now I'm going to segue and go into the idea of where I came up with this talk and see if this makes sense to people. I'm sure it does, but it did to me at the time. So there's always been a book, it's not always, there's a book called A Feeling for the Organism, and it's the life of Barbara McClintock. And so she was a very famous geneticist at the turn of the century at a time when women weren't really doing science. And so she did beautiful studies to understand the genetics in corn and other plants. And she basically discovered transposons and won a Nobel Prize. But from the book, I'm going to just kind of read this here. So what enabled McClintock to see further and deeper into the mystery of genetics than her colleagues? Her answer is simple. Over and over again, she tells us one must have the time to look, the patience to hear what the material has to say to you, the openness to let it come to you. And above all, one must have a feeling for the organism. One must understand how it grows, understand its parts, understand when something's going wrong within it. An organism isn't just a piece of plastic, it's something that is constantly being affected by the environment, constantly showing attributes or disabilities in its growth. And so I'm not Barbara McClintock, so there's not a feeling, but I maybe have an inkling for the organism. And there's something that's beautiful about studying this worm that gives you a lot of clues about behavior and function. So when you look at mutants, you saw that elegant wave. If it sort of loops, I know the nervous system is hyperactive. If it's kind of flat, right, I know that it's not working properly. If it's holding too many eggs, I know something's wrong there, right? So just by looking at it, you get a lot of clues and understanding of things and behavior, and you really get to appreciate that. And as I'll talk and mention in the talk, there are examples of students kind of making those same observations. Okay. So a little bit more about the background of the worm and why it's a model organism. I should say it was discovered by a scientist, Sidney Brenner, in the 60s, and he found it in the soil. So it's just found like in the orchards of Washington, you would find it. It's just where there's compost. So it's not parasitic or anything. You can see that it's transparent. So you really can see all its structures. So one of the things is it's a self-fertilizing hermaphrodite. So these are, it says ovary, but these are the gonads. So what it does is it makes a bunch of sperm first and keeps it in something called a spermatheca. And then the oocytes, right, eggs develop. And when they kind of go through the spermatheca, the eggs it becomes a fertilized egg. The sperm and the oocyte make these eggs. So a single animal can make 300 progeny, right? So it's really easy to maintain. I don't have to mate or cross it, does it all itself, right? And it grows from an egg. You can see, I don't know if all of you, I'm not going to try to move anything. I'm gonna break it. From an egg to a full adult in 3 days. And you know, I was passing around plates before. Hundreds of them are on these small little auger plates with a lot of bacteria for a food source. So they're pretty easy to maintain. And so it's kind of the perfect organism. It also has all of its cell lineage identified, so we can see it transparent. So when it divides, we know what every cell becomes, what tissue, and especially the nervous system. So we know all 302 neurons in there, and then we also know all the connections that they make. And so it's much simpler with 302 neurons versus 100 billion in the human brain. Plus, you can't really do experiments on humans. Other limitations. Right? So again, another thing making this worm quite attractive. Okay. So the other thing that I sort of alluded to is it also has genetic tools, so we can manipulate it really easily. So we know it has about 20,000 genes, which surprisingly is about the same number we have. They're not the same genes, but about 50 of them are similar to human disease genes. So that's what we mean by ortholog. So there's like worms have a human version or humans have a worm version, however you want to think about that. We can make mutants really easily. We can put genes into them. We just inject DNA into the gonad, and then the oocytes and the sperm just kind of collect it, and it ends up in the progeny. We can do genetics, so we can screen for specific defects of whatever we're interested in studying. And then reverse genetics is we can also specifically target certain genes. So if there's a gene associated with a disease and we want to see what that function might be, it's really easy to knock that out and see how it goes. So now I'm going to kind of transition. And so, oh, my story's long. No. So I worked before I came here as a research associate at a place called the Ernest Gallo Clinic and Research Center. And what they studied is alcoholism and drug addiction. And so I used that worm to study the effects of stimulants like amphetamine, cocaine, heroin, pretty much those things. And so the pathways that they affect are the neurotransmitters, serotonin and dopamine. So when I came here, I decided I probably still didn't want to use drugs that require licenses and things like that. I still wanted to study those systems. Plus serotonin. So I should say those drugs of abuse affect serotonin and dopamine. So those are kind of the neurotransmitters I've still been looking at. And so in particular, I've done a lot of work looking at serotonin. And serotonin is an interesting neurotransmitter in the brain. You can see it's got pretty much all over the brain, and the nuclei are right kind of in the brainstem there. And it regulates important behaviors in humans, right? Appetite. So it controls us, or it helps us from not overeating. It also does the same thing in the worm, so that's analogous. Mood, we know that if we alter the levels of serotonin, it helps treat depression, right? And then there's aggression. It helps regulate sleep or arousal. And then, like I mentioned before, serotonin plays roles in addiction to drugs of abuse. And so not all of these things we can study in the worm. I can do appetite. Some people can do sleep. I don't really do that. And we can't really do mood or aggression, but there's kind of a way that we can indirectly look at addiction from a cellular, like a nervous system level. Okay. And so in the worm, there are only 8 neurons that make serotonin, and they're shown here. They're kind of lit up with this GFP protein. So there's some that regulate the rate of feeding. There's some that are important for their slowing in response to food. And then there's some that regulate the rate of egg laying. And so we know what behaviors are associated with those neurons. And so changes in those can be measured by looking at these different behaviors. And so when I came here, so I should say those 3 behaviors, there are ways to kind of quantify them, but there weren't really good ways to quantify locomotion. And so when I came to the lab, I had a colleague working at Stanford, and I spent a summer down there and learned how to use this software tracking program. And so this allows you to kind of measure things in a more objective way. You know, looking at egg laying where you're determining with your own eyes is a little bit subjective. And so what you can do is you can transfer worms onto a plate, and we call these corrals. They're made with paper, and we basically put a repellent on those so the worms stay in these little regions. And then what you can do is put them on the microscope, and then here we have a camera that goes to the computer, and this program identifies all the worms based on size, and it makes these tracks when you record. So we record for, say, 15 seconds, and these tracks represent the distance. So distance divided by time gives you speed, so we can measure the speed of these worms. And so this was a project there. We first started developing this. So a lot of times in working with neuroscience and anyone who does that in any organism, it's always trying to find a really quantifiable behavior to measure what you think you want. So part of your work is really just designing the experiment to do the experiment, which I think has actually been kind of some of the funnest parts of doing it. So here I'm going to mention Eric Foss. He was, when I first got here, he was my first undergrad student and also my first graduate student. And he worked in the department for many years. So he had a lot to do with kind of initiating and staying interested in this project until he found another organism he liked better, which were like yeast, which, you know, you can use for brewing and things like that. You guys don't know, Eric's the brewer at Ellensburg Brewery. So here's what we measure. So the speed of a worm that's not on anything goes at about 250 microns per second. And so as we add increasing doses of serotonin to the plate, you can see that there's a decrease in speed. And you can't really see here, but these are worms on serotonin. And you see they're not really moving. So that's what they look like on serotonin. And in fact, they tend to want to clump. That's another thing. They want to hang out together. And so, but I just wanted you to know we can also just visually see this too. So that goes back again to the feeling of the organism. So often when experiments don't work, I go back and look at the videos to see if everything looks right. I always say, go back and look at what did you see? Right. So we wanted to use this to show we could measure changes in serotonin. But what I was really interested in is something called behavioral plasticity. And so that's the ability of the nervous system to modify its behavior due to environmental changes, and it's usually over time. And so some of these sensory modalities that you might be familiar with, it's like if you've been in the dark and you step out into the light, it's really bright, and then you adjust to it. If you drop a paper on your hand, you initially feel it drop down, and then you forget it's there. Like if you close your eyes, you wouldn't know it was still there, right? And so you're adapting to that sensation. And then what is not a natural sensation is we have a form of adaptation to, say, food and drugs, and that's kind of seen as like a tolerance, right, to chemicals. And that's a form of adaptation. And so that's what I was really interested in, is how the nervous system adjusts over time to these different stimulants. So it's like a change. And so we developed a way to look at that. And so I mentioned here that there's an initial exposure to serotonin. You saw how slow those worms move. And so we wanted to know if we treated them overnight on serotonin, what could we observe? And so if we take these that were on serotonin and we keep them on serotonin and measure, can we measure adaptation? Are they changing over time to the initial response? And then the last one is sort of a withdrawal effect, which you guys might be familiar with in terms of drugs, about why you don't want to abruptly, even if it's medication or drugs, you don't want to necessarily withdraw immediately. So here's the same movie where they're not really moving. So this is an acute. And the bottom one is the worms that have adapted. So this is this protocol here. So you can see that they can recover visually over time to the movement, right? And so we have now found a way to look at adaptation. And if we graph that data, right, this is the acute, you get the slowing. So this is what the speeds look like of adaptation. Although they're not the same as the control levels, you can see they're clearly moving, so they've recovered, right? And so that's what we call adaptation. What Eric was also able to find is that there was also a withdrawal effect. So often withdrawal is thought of as having the opposite effect of the acute response, right? And so that's why withdrawal symptoms are rather unpleasant if you're thinking about it in terms of drugs. So we could actually measure that they're going faster when we remove them from the serotonin. So it's kind of a rebound or withdrawal effect. And so we showed that we could identify all these different behavioral responses to the work or to serotonin. And so the next thing we wanted to do, right, is we wanted to figure out what genes or proteins might be regulating this, right? How is adaptation occurring at a synapse? So there's probably changes in proteins at the synapse, right? And it's the connection between the neurons, right? So they're becoming more or less responsive. And so what we did is we did a genetic screen to look for animals that failed to adapt to serotonin. And so you can give them a chemical mutagen that causes mutations in their DNA, and it's sort of random. And then it takes 3 days for one generation to grow up. And then we let the second generation grow up, and there's kind of genetics, but you can get different mutations if you look at this generation or this generation. And so we basically did both. And then what you do is you look for ones that fail to adapt. And so what that would look like, right, is when the student did the screen, is that there was a recovery of speed normally. I didn't write. So if they fail to adapt, they don't recover. So they look just like the worms that were acutely exposed. So we look for worms that couldn't move. And so literally there are different ways where you just screen for the ones that could move and you pick the ones left behind. Okay. And so, and that was done by an undergraduate student, Spencer Moen. And so this was the title of the source talk. So in C. elegans, we like to try to be cute. So the speed of happiness is sort of misnomer about what serotonin does. But anyway, so I let him go with it because, you know, it's their project, so they get to own it. And so wild type on a food, so he noticed a phenotype of these worms even when they weren't on serotonin. So normally they spread all over the plate. They're considered solitary. And so what happens with these worms, it's sort of hard to tell, but this is the lawn of bacteria. So that's their food source. So these animals all move together, and as soon as they hit the lawn, they stop. And then they eat more food and they move. So they are not solitary. They're very social, but he described the phenotype, the defect, as depressed foraging. So they weren't foraging all over for food. They were just getting the minimum amount. And so in C. elegans, we name a mutant based on a phenotype. So we get to describe the phenotype. And so it became DEF1 was the mutants that came out of the screen. And so this is the first one called DEF1. And if you look, then if we look for the effects on serotonin, we notice that the DEF1 moves slower than the wild type animals. But then when we give it serotonin, the acute response, it's sort of extra sensitive, right? It's slowing down even more. But as you can tell, the wild type, right, recovered their speed and these guys didn't. And so we can measure and confirm that defect in adaptation. And so one of the other things about the mind of the feeling of the organism, or in my case, the Inkling, I was hanging out in my friend's lab and I was looking at a mutant called PKC1, doesn't really defect in a gene. But I also saw it had the depressed foraging phenotype, right? And so that gene is part of a pathway within the cell that signals whether neurotransmitters get released. And so I was able to identify a whole pathway that was involved then in that adaptation to serotonin by just making that observation of those worms off serotonin. So that's what I need. Sometimes there's just Inkling, you're just playing with the worms, hanging out, and things come up. So, yeah, I do. And, yeah. So then the other thing after this, like as humans, we're not bathed in large amounts of serotonin, right? Those are kind of abnormally high levels of serotonin, although they are high because the drugs don't always get through the cuticle of the worm. So we have to put high amount, but we don't know how much actually gets in the worm. But what I really wanted to look at is a more applied application. And so I decided that I wanted to look at drugs that elevate serotonin at the synapse in a more natural way. And those are the selective serotonin reuptake inhibitors that people use for treatment of depression, right? And so what happens is that these antidepressant drugs block the transport. So when we release neurotransmitter, it's a quick signal, and usually the cell takes it back up and puts it back in the vesicles. And so SSRIs block that, so you have more serotonin at the synapse. And it's believed that people with depression just don't have enough serotonin at the synapse. And actually, it's a little bit more complicated than that. I think actually having extra serotonin just helps with the depression, and it's not really the cause of depression. And so here's some examples of these antidepressants you may have heard of: Paxil, Prozac, Lexapro, Zoloft. So these are all ones that target this. And this work was done by a graduate student, Maria Nemeth, and an undergraduate, Taikeya Baird. And I had Taikeya for all 4 years when she was here. She was part of the STEP program. She ended up doing the Science Honors program as well. And yeah, so I spent a long time. So we wanted to see if we could mimic the effects of antidepressants in the worms. And so the way we do that is what naturally releases serotonin in the worms is food. So they're on that source of bacteria. And so when there's no, when there's a plate with no food, they move faster, they don't lay eggs, and they don't pump. But if there's food, they're where they want to be, right? So they're going to move slower, they're going to produce progeny, and they're going to feed, right? And so what we did was we put the antidepressants and food on the plates with the worms. And this is the result. So if it's similar to serotonin, so on food, they go slower. That 250 was off food. So food slows them down initially. And then if you add serotonin, the speed slows down more. And then this is one of the antidepressants. So we basically screened a bunch of antidepressants. So this seems to act like an SSRI because it slows down, right? It increases serotonin, so the worms slow. The worms slow here, they slow less here. And if you notice for these three antidepressants, this is Cymbalta, this is Paxil, and this is Prozac, the opposite is happening, right? They're not actually slowing down, they're speeding up. So what does that tell you? No one knows. Okay, well, for me, I was very excited. That's the inkling for the organism, right? So it means that it's acting probably on something else. And so we have side effects with a lot of antidepressants, right? Or a lot of drugs. And it's because they have other targets that they affect. And so part of Maria's thesis, which I don't have time to go over, is we identified the other target was a dopamine receptor that it was inhibiting. So now we know these antidepressants maybe affect other things than these SSRI. And so that's something that this can kind of tell us is what might be responsible for some of these side effects. So, oh yeah, so that kind of concludes that one bulk of my story. And now I'm going to talk about a couple of projects that were really student generated. And so one nice thing about the worms is it can be applied to a lot of different systems and areas. And so I had about the same year I started, one of my first students in my biology 110 class was this gentleman here, Steve Chrisman. And he was originally just going back to school to get enough credits to be a paramedic. And then as he started taking biology, he kind of fell in love with that. So he ended up getting both degrees. And so he comes to me after he'd had me for one class, the first class I taught. And he's like, can I have the worms? Can I have the worms? I want to do this thing. I want to look at what they do in electric field. So there's a 9-volt battery and a little auger plate and I don't know, some wires. I was like, yeah, whatever. Electro what? I don't know. Yeah, I can give you some worms. They're cheap and easy, right? And I don't know if you remember, I know Dr. James remembers because he's very excited about his project. So this was an animal biology project for Dr. James biology class. Right? And so that was kind of how that started. And then there was a journey. And so he was very persistent in this. And so it got a little more elaborate. And so that's the system. He refused to give up on this. So we got a big old power supply that could probably electrocute more than worms from a physics professor, Michael Jackson at the time. And then he built this apparatus and we had to go through all these things because you heat up, right? In electric field, you try to do that. You generate heat, you generate ions. Like he spent forever with buffers. I smelled chlorine in the lab one day. I think we're dissociating sodium chloride and we're like, chlorine's not that bad. So, you know, but he kept going with it and he ended up with science honors and a master's. And so, yeah, he had it all rigged up. And his very first ones were on here. And he would monitor it and we would tape it with the VHS. And so it was not sophisticated technology at all. And in fact, he never saw the monitor when he was playing. And one of the observations with this electric field I'll tell you about is when you turn it on, they move very directly. When you shut it down, they actually reverse. And so I remember the day he was playing with everything. I'm like, did you see they reversed when you turn that down? And so that's another thing about the inkling of the organism. And that became part of this project, right? And so I finally discovered, right after he'd spent years working on this, and I needed to get something out of it. So we both did some reading. And it turns out it's really an interesting sensory behavior that is quite common throughout organisms, right? So it's a very conserved behavior. So sharks detect electric field of prey, right? So that's how they find them. Same thing for the platypus. These are fibroblast cells. So during wound healing, the fibroblasts go to the site of injury. They sense an electric field. So they move to the negative pole of the electric field. And then this is dictyostelium. They go from these slugs to these stalk structures, and they sense electric fields to make that transition. And so in the tip of the worm, you can't really see it, but there are little holes or openings outside the mouth. And it's called an amphid, and that's their sensory organ. So that's how they smell, it's how they taste, and now it's how they kind of detect electric field. And so there were old papers that people had looked at this electric field behavior, but you know, it was with the 9-volt battery and some wires. So I don't know how much they really could find out about it. And then if you look at the opening, there are nerve endings that are exposed to that opening in the amphid. So there are neurons that are amphid neurons. And so by the end of this is kind of cool imaging. He was able to, with the help of another undergraduate, kind of modify that worm tracker program to really be able to track and analyze this data in an interesting way. So this is the electric field. This is going in the negative pole. So this is no voltage, then we increase the voltage. You see, these are tracks of worms. So you see how they're orienting and they're moving. You can't see it, but in the movies, they're moving very quickly towards that point of the negative. And then when you go up in voltage, they start angling. And this is just kind of a cool way to represent this. This is the angle at 1.5 volts. You get that really clean peak, and it starts to spread out, and then it becomes 2 peaks. So in this little auger pad, he had a bunch of pins to measure the electric field in all these different points in an array. I don't even know how he did it. It's like hundreds of, I wouldn't have the patience to do that. But he did that at every angle with the field strength. And so what we realized is that the angle, so as you increase the field, it's moving, right? The field's too strong, so it's deflecting away. And as you go up, it deflects more. And so if you calculate, the worm is always oriented, so it's moving at 4.2 volts per centimeter. So it has a very specific voltage that it's recognizing. And if it's too high or too low, it tracks away from it. And so that's really kind of interesting. And so that's kind of, and then the last thing, I'm just summarizing this paper, is that we were able to find this is a neuron, one of the amphid neurons, it's AWC. And so there are kind of 2 forms of it, but you can see this one moves normally, but when we have a mutation in it, you see that it loses its ability to sense that field and disperses everywhere. And so we identified probably the neuron that's also responsible for detecting that electric field stimulus. And that ended up with this paper here. He's the first author, and it's pretty neat. It's got a lot of reviews and some citations. So it's kind of one of the big papers for looking at electric field stimulus in C. elegans. And it was kind of interesting. About maybe 6 months ago, there was a physics lab that has a way to look at imaging of neurons in the worm that get activated, so they can see the nervous system activating in real time and what neurons. And they wanted to do repeat his electric field apparatus to see what neurons were lighting up during the response to the electric field. And so that was pretty cool. So that was all student generated. And then another one is kind of another student generated project. And so C. elegans can actually be used to study Parkinson's disease. So what happens in Parkinson's disease is these particular neurons that contain dopamine die, right? And that affects your motor coordination. And so in the worms, we also have dopamine neurons. And so you can visually see, because they're transparent, if there are things that could cause these neurons to die. And I had a colleague at the University of Alabama who gave me a strain because he works on this. And this is a human protein that when that is put in those neurons causes early Parkinson's. And so this is a model for Parkinson's disease. And this just shows you in C. elegans and now in every system, there's this green fluorescent protein. So this gene we can put into worms so we can really nicely visualize cells because the worm's transparent. And so they're just primarily, we usually look at just these 4 CEP neurons. And so you can just see them in the microscope. You can have one of the fluorescent microscopes going there, and those dopamine worms. And so the student, actually 2 different students. So this was originally started a long time ago by one undergraduate I'd forgotten about, but the most extensive studies were done by Jake Darley, who was an graduate student in science honors, STEP, all these programs. So was involved in undergraduate research. And so they wanted to look at, in plastics, right, what makes them malleable are these compounds called phthalates. And we know that they can leach out of the plastic and they can have detrimental effects. The most common one is BPA, probably heard of that, and that affects reproductive systems. And so they wanted to see if these compounds were affecting the degeneration of, causing degeneration of the nervous system. So I should say Parkinson's disease, a very small percentage of it's genetic, but most of it is just developed and we don't know why. So probably some kind of exposures over time. So basically this is sort of what this looks like. So this is a little bit overstaining. You see these really bright, healthy neurons. And so if they're undergoing degeneration in Parkinson's or with treatment, you see that we lose them. So now we only have the 1 neuron here. We've lost the other 2 over here. And then you see these processes start to bleb. So those axons are breaking all up and dying. And so this is how we assess degeneration of these neurons. And so when Jake looked at one particular phthalate called BBZP, what we saw over time, so this is looking at degeneration of the neurons, and this is just the agar. We make the compounds in the solvent, so we use that as a, we call it vehicle control. And then you could see at the higher doses, we got an increase of degeneration for the worms with the phthalate. And so now we've done other studies looking at these phthalates in mammalian tissue culture. I've got projects involved in that. And that kind of is an example, another of a student-initiated project. And I should say my colleague looking at Parkinson's in this model, they've actually helped identify drugs that then prevent this, right? And so the worms can use to screen a lot of drugs the target. And so those neurons have the same receptors, the same sort of pathways for the synthesis and function of dopamine. And so they really are analogous. And that's been shown in different systems, things we find in worms or in mice and so things like that. And I think just to finish up, this was a project a while back. So this wasn't started by a new student, a novel project. It was started by a colleague who I call a mitochondriac, and that's Corinne Thomas. And so a mitochondriac is someone who loves mitochondria, which is the powerhouse of the cell. And so she's a biochemist, and that's what she thinks about all day, every day. And so she came to me with a student, I guess it was a student project, that wanted to look at the effects of high-fat diet on mitochondrial, what stress that would put on cells and mitochondria. So that's one model for type 2 diabetes, is that high fat diets can stress out the mitochondria and it causes damage, and then those cells can't secrete insulin. And so we actually were funded with an NIH grant. So this is only a few of the students, but over the years, we had a lot of students that we could put on this project and work on very different aspects of it. And so this is sort of a picture of an example of what you could look at. So this is a dye that when the mitochondria are producing what we call a reactive oxygen species, which is a form of stress, and this fluorescent dye, these are mitochondria in the head of the worm, they increase. It's a measure of that damage. And so you can kind of see that we can do that. Corinne was able to put the worms in a little test tube, or the students were. We can measure ATP levels. We can measure oxygen consumption in these little things, lipid peroxidation, all damage, all evidence of kind of cell stress. And so we did all those things, and I'm just going to kind of summarize. This was a grad student of mine, and so one of the things we found is that when we give them these high, so there's saturated fat and unsaturated. Saturated is supposed to be the bad fat, right? And then there's unsaturated. And then this was just something we know causes stress. But we could see this protein light up in the pharynx of the worm, which is where they pump food. And this protein is a protective protein of mitochondria. And so what Maya was able to find out is that the exposure to these high-fat diets was causing these protective proteins to become upregulated. And so all those other experiments, we didn't see evidence of damage. And so what we realized basically is some stress can actually be a good thing for the cell, right? Because it activated these protective mechanisms. And so this made it a little bit hard to publish this because most of the results were negative. But anyway, so that's kind of a story. And I think that's where I finish off. And I just want to acknowledge every mentor and colleague I ever had and every student I had an opportunity to work with. This picture is the one on the website of me. I always cut it in half, but this was a fun day that we had to take a picture. I had to put on a lab coat. She didn't have a proper shirt on. We were getting photographed because she had won from the Genetic Society of America Travel Award, and they wanted a press release. And so the only thing we could find, because we never really wear lab coats, I think one of the students had it left over from chem lab. And so she put that on, but we were kind of laughing and having a good day in the summer lab. And then this was my my postdoc advisor in worms, Marty Chalfie. And so he's well known because he discovered how to make transgenic animals with that green fluorescent protein. He figured that out. And sometime after I left the lab, he won a Nobel Prize. And so I think this was after this, and we were at a worm meeting. This was Eric and Steve, and they really wanted to meet him. And so I kept trying to stall him whenever I ran into him, but I was never with those guys. And so the whole meeting, they just kept missing Marty. And then he got locked out of his dorm at this meeting at UCLA campus. And so it was like 11 o'clock at night. And we were coming back from the poster social hour. And so we got our picture because had he not been locked out, we would have never. And then these are my current projects. I've been kind of getting back up and going after COVID and sabbatical and being McNair Scholar. And so Hannah's gonna will be a grad student working on intermittent fasting and neurodegeneration as a protective mechanism. And then Ricardo and Edward are kind of have been working on generational effects of serotonin and SSRIs on behavior. So to see if these changes can be transferred from one generation to the next. So that's it.
Questions? Is everyone's question? We really have.
Want to work on worms.
Yeah. I have I was always so many questions. Kind of thinking about like the way that this intersects with sort of like students, kind of interests and what they then go on to do, like in terms of your students, like, are you seeing a lot of these students who are going on to continue to work with worms, or are they working in other areas with other animal model situations, or
so many things? A lot of our biology students end up going into health fields, so that's where a lot of them end up. So, but this experience gives them good experience just about work experience. It helps them, I think, even understand how to think as a medical practitioner, right? To kind of really understand these diseases, probably at levels that they never would. So Chris and Luis, their daughter, who went to UW, showed up one summer in my lab and wanted to work on worms. And at first I thought they're kidding. Chris was like, yeah, my daughter wants to, not even from here, right? Wants to work in my lab. And I said, okay, sure. And so I spent the summer, but Joelle works on worms and went to Rutgers to get her PhD and just finished her PhD, right, in doing C. elegans, and worked at, so my advisor, Marty Chalfie, I'm getting this all backwards. So Joelle's advisor also worked in Marty's lab. So we were kind of like, we weren't there at the same time, but we were like academic, siblings. Step siblings maybe or something. And so anyway, but that had an opportunity, and she always talked about how that helped her understand, when she went to work in a worm lab, she felt she had a little bit. But yeah.
I'm always curious because I know in anthropology, a lot of times students come in with an interest that's kind of related to like their identity or who they are, right? And when a student works with something like C. elegans, which feels really different from humans, I'm always interested in sort of like how it is that they find that sense of resonance with something that seems really different from them.
Yeah, so they know I work on the nervous system, so a lot of students are very inherently interested in that for different reasons. Some of these diseases we talk about affect them personally, and so that's one of the things I usually ask students if they want to do research with me. I say, well, why my lab? And it doesn't have to be some brilliant thing, but it just might be as simple as that. So if there's some connection, it does help, but some I also take because they don't know and they just get amazed. So I use the worm a lot in my classroom. So we've done projects to test novel theories about how to protect against Alzheimer's. I had them help map that DEF1 gene in when I did molecular biotechnique. We did that as a project. So they're actually doing real science in the classroom.
Yeah.
So
Any other questions? I guess if you have a bunch of scientists in the room, there's not a lot of questions.
They've heard plenty about my worms. That's all I talk about.
Enough about your work.
Yes, I've been around for 17 years.
So I'm not sure if there are any questions on YouTube because I'm trying to toggle to YouTube, but I'm not. Let's see.
Where do you get your worms that you use? Do you order them from someplace?
You actually can. So there's a center repository in Minnesota that keeps a lot of the strains, and for $10 you can do that. So when people publish, they send some of their mutant strains. But yeah, the standard wild type one you can just get. But yeah.
So one question from YouTube was why worms?
That was in the top. That was that slide. Very simply, it has a really well-described full nervous system, and I can really manipulate it genetically, so I can identify genes associated with a lot of interesting behaviors.
Mm.
And it's really easy to maintain. I don't have to mate, right? So, and these things, I don't have to mate it. And like with flies, people always have to make sure they come in and mate them. And these freeze, so I can keep them. They'll go into a starvation state. So as long as I pair farm that and it doesn't dry out, those can go for months. So I can go on a long vacation. I can take summer and I can come back and just put them on fresh food and they start growing. They don't fly away like the flies. I don't need any kind of animal protocols, right?
Oh, so there's no Ayacook for worms. I never even thought of that.
There's
Yeah.
no
I
what?
literally can take these on a plane. So if I go to my friend's lab and they have a couple strains I'm interested in, I just grab it, parafilm it, put it in my backpack, and
Like human subjects review, except for animals instead of for people.
yeah, like,
Yeah.
Let me try to articulate this. So I think this is fascinating in so many ways, right? Thank you for all the work that you have done, and especially with students. So for me, as in terms of philosophy of science, you have this challenge, right? You are using microscopes to do these tiny little creatures and identify and invite students to see, to render visible what is not with these tools, right? But in terms of, especially as McNair and serving underrepresented students, when did you find that seeing these things that we don't see on a daily basis allow students to see themselves as scientists, things that maybe they haven't conceived?
Well, I think
That journey.
so. It's really easy. They're really pretty quick to use. So like I can do a lot of these investigative experiments in a classroom, so they don't need a lot of expertise. But the tricky thing is there's a little metal platinum wire to you. You have to pick up individual worms, so you can't do that after you've had a big cup of caffeine. And so once they learn how to pick, I can get them to pretty much do anything. So, and we do a lot of standard molecular biology that you would use in any organism, and we can do that as well. But yeah, it's just it's easy. I can get them going on a project with very little training or supervision. So, and they can really think, I think, really kind of big questions and really get a feel for even just kind of science, sometimes just basic science too, right? Just that's what I mean by the inkling of the organism, right? So yeah. Yeah, they do all kinds of other weird behaviors. I could go on and on.
I wonder about electrotaxis in humans. Like you talked about the way that
The
our
cells respond.
yeah, when we're healing or whatever, but like I don't know. I mean, it's not, obviously you can't electrocute humans for this to happen.
So this is
But-
below this,
yeah.
right? That causes
Yeah.
So Steve could crank it up and they flatline and become paralyzed, and then
Yeah.
you could reverse it, right? And that's a much higher field strength. And so
Yeah.
So do humans have
Like, do we move and respond in ways to electric fields that we're unaware of?
well, we're electric, right? So
Yeah.
I mean, I sound like isn't that a poem? Where's Michelle? Where's the provo? She always knows her poem. I feel like that's I am the body electric. Okay. Well,
Yeah,
anyway,
that's-
so all living things have a polarity and a charge across their cells. So
Yeah.
who knows? But the other organism are actually bees can detect different
Yeah.
flowers to pollinate by detecting that field.
I had heard that before.
I haven't looked so much following humans. I haven't kind of kept up on this.
Or non-human primates.
Yeah, sorry.
You don't know? No. Anyways, that's interesting to me.
Platypus is almost a mammal.
Almost.
I mean, very super close, right? Super close. Yeah. All right. Any more questions? Look at YouTube real quick. All right. Great talk, Lucinda. So, thank you so much.
With the fluorescent scope, if anyone wants to see some glowing worms.
Definitely. Please check out
Before-
worms.
before
Yeah.
someone has